neural reconstruction here means utilizing surgical procedures to reroute a healthy peripheral nerve to connect it with a paralyzed peripheral nerve distal end to reanimate motor sensory functions. Previously, my lab has published this paper in which we tested feasibility of borrowing a healthy nerve. Here is thoracic 12 nerve that is above a thoracolumbar ipsilateral hemisection injury at T13L1 spinal cord to reroute it with the paralyzed lumbar 3 nerve. When we did this 1 week after spinal cord injury, as showing in this locomotion score evaluation for these animals, we found that the green bar, which are the animals received such a procedure within a week to 9 days after spinal cord injury, they all showed very robust functional recovery in locomotion for the affected hind limb. Here is a piece of video that you could appreciate that how this ipsilateral injury could completely paralyze the affected sides, resulting in permanent functional loss. With this procedure, we were able to recover that function. But if we look at the dark bars, those are the animals received this procedure 1 month after spinal cord injury. They did not show any recovery, imposing a challenge to applying this strategy for a chronic stage of spinal cord injury. And in those groups, we also found that if we put a nerve growth factor called FGF2 into the paralyzed muscle, the target muscle, that the rebuild reconstructs the nerve root are going to reinnervate, we found it can further enhance such functional recovery, suggesting that this reinnervation process is important, is very important, but also perhaps there are other neuroplastic events occurring within the spinal cord to reanimate the locomotor generator circuits. Now we have focused our efforts on testing the same strategy after contusion injury. The contusion again took place in thoracolumbar segment, which is the second most frequent places injured in humans. When we done such a nerve rerouting, again, within the first 9 days, we again see a sustainable, this red line, functional recovery, this time from both legs, because this is a symmetric contusion injury. But if we wait for 8 weeks, the blue line, despite the nerve reroute surgery, the animals did not recover. We further explored a possibility of using stem cells to rejuvenate the late phase spinal cord after trauma. We injected these cells 1 week before such a nerve transfer procedure. And in the following week, at week 8 after injury, we did nerve transfer. And once again, the green line showed you the combined treatment can induce functional recovery. This is very exciting because we think we might figure out a pragmatic way to apply nerve transfer, which has known efficacy to chronically injured spinal cord. We're now concentrating on analyzing the key mechanisms that support such a reactivation of the lumbar locomotor generator circuits, involving the neuromuscular junction repair, the outflow of this borrowed motor neuron control to the paralyzed muscles. And more importantly, as these other arrows showed, what are being sent back through sensory nerve to the lesioned spinal cord. They can work in consortium to reactivate pattern generation, to reanimate both hind limbs.